Good morning, student. नवयुवक शिक्षण प्रसारक मंडळ चापोली संजीवनी महाविद्यालय चापोली welcome you in e-learning platform prepared by myself Dr. Shubhangi Prabhakarao Chavan, Assistant Professor and Head Department of Botany. Now, Swami Ramanand Tirth Marathwada University Nanded Semester Pattern Curriculum under Choice Based Credit System CBCS for Undergraduate Program, Faculty of Science and Technology, Subject Botany, Year BSc Second Year, Semester 4, Paper number 8, Plant Embryology. Now, student, in last period we discussed about syllabus of 4th semester. From today we will start paper number 8, Plant Embryology. In this paper, unit first, Embryology. Let us start Embryology. Definition. Embryology is the branch of biology concerned with the development of new organisms. Embryologist takes reproductive cell gametes as they progress. The fertilization become single called zygote, then an embryo, all the way to a fully functioning organism. They are mainly subdivisions of embryology. Some scientists focus on human embryos while others study animal and plants. Now, next, evolutionary biologists often use embryology as a means of comparing species, as the development of an organism can give many clues to its evolutionary history. Embryology is the study of the structure and development of embryo, including the structure and development of male and female reproductive organs, fertilization and similar other process. Father of Indian plant embryology, Panchanan Maheshwari. Plant embryogenesis is a process that occurs after the fertilization of an ovule to produce a fully developed plant embryo. This is the peridient stage in the plant life cycle that is followed by dormancy and germination. Now, next, the zygote produced after fertilization must undergo various cellular division and differentiations to become a mature embryo. An end stage embryo have few major components, including the shoot apical meristem, hypocotyl, root meristem, root cap, and cotyledons. Unilike animals embryogenesis, plant embryogenesis result in an immature form of the plant, looking more structure like leaves, stem, and reproductive structure. The dendrograms are also called spermatophytes. The plant propagate mainly through seeds. The seeds is a structure in which the embryo is enclosed. adjacent to the embryo. Next, role of embryology. First of all, dicot and monocot. Angiosperms are usually divided into dicotyledons and monocotyledons. The primary classification of angiosperms is based on one major embryological characters, that is the number of cotyledons. Next point, halobi. This is monocotyledons order treated as a subclass in some recent system of classification, is characterized by the presence of holobial type of endosperms. Next point, orchidales. The distinguished embryological characters of the membrane of this order is the presence of undifferentiated embryo and very little or no embryo endosperms. Now, next point, Podostamesis. Members of this family are recognized because of the formation of pseudo embryo sac, which is formed by nuclear cells. Next point, Lamiaceae. Phylogenetic studies indicate that Lamiaceae have been derived either from the halobials or from the aerials, but on the basis of the embryological studies, Maheshwari suggested that Lamiaceae have been evolved from Ariaceae, not from halobials. Next point, Crucilaceae. Embryological studies of Crucilaceae suggested that it studied be placed in the order Roselles. All the above mentioned examples confirm that embryology play a definite and significant role in solving taxonomic problem. Now, next we will discuss in short life cycle of flowering plant, alternation between the dominant sporophytic generation and a highly reduced gametophytic generation. Dominant sporophytic generation is diploid and reduced. The normal sexual cycle involved in two important processes. First of all, meiosis and fertilization. Next, fertilization. In meiosis, also known as reduced division, a diploid sporophytical cells, spores mother cells, get converted into 4 haploid gametophytic cells. 2n number of chromosomes become half, that is n number of chromosome. Gametophytic generation is haploid. Now, here is diagrammatic representation of life cycle of flowering plants. Now, we see typical flower in that we observe androecium and gynoecium. Another, the produce stamens. From stamen, pollen grain formed through microsporangium. Here we observe microgametophyte N, that is germinating pollen grain. It is ready for pollination. In other side we can see gynoecium, through which megasporangium formed and it produce megagametophyte N. Then mature ovule forms at one point between mature ovule and microgametophyte, pollination occurs and new embryo developed in seeds. Further, it developed into young sporophyte, that is 2 young. Again, sporophyte reproduced the flower. Thank you, student.